So we all want to make good models, and I think that, in my opinion, you can reduce a good model to certain decisions that you make in the process, and they're outlined here. One is segmenting the tissues correctly, so whether you're separating out brain and CSF and so on. The second step is applying the correct properties to the tissues. So typically, we're assigning them resistivities, for example. For electrical stimulation modeling. And so ensuring that you assign cerebral spinal fluid an appropriate resistivity versus skull a different appropriate resistivity. The third step of it is correctly modeling how the energy is directed into the body. So modeling the electrode for electrical stimulation or the coil in a way that is meaningful and captures the appropriate properties. And finally, there's a step of how we actually represent neuromodulation. Which can be as simple as plotting the electric field in any given region, or can become more sophisticated, even going all the way to incorporating neural models. And I think it's important to recognize that to some extent, all models are bad. All models omit major details that are possibly very important. And so when we talk about what makes a model good, I think it's very important to keep in mind that it comes down to what the model is used for. And if the model is being used to answer a particular question, these steps, 1, 2, 3, 4, should be selected in a way that allows us to answer that question appropriately and meaningfully. So we can start with one type of model, which is concentric sphere models. These have been around for decades. And they are, one sphere inside another sphere representing different tissue compartments. So the outer sphere, for example, would typically be skin or scalp, and then you'd have inner spheres representing skull, eventually brain. And being spheres, they obviously don't represent any form of realistic tissue anatomy. But I'm going to make the argument that for exactly that reason, they're useful, meaning they are good to answer certain questions, right? What question you want to ask, because they don't have anatomy. So we and others have been modeling spheres for a long time. This is a paper from 2008, and here the specific details don't matter except to say that we're modeling different montages, two adjacent bipolar electrodes, a tripole, a belt, and then this notion of a center electrode surrounded by a ring. And these are all concentric sphere models. This is using finite elements. You can see the mesh, and we predict the electric field. And where are we predicting the electric field? On the sphere. So there's different spheres representing skin, skull, CSF, and brain. In this case, brain is one compartment, and we're predicting the electric field on the surface of the brain, because that's where we think the neuromodulation happens. So there's that notion of where we represent the electric field, not on the skull, in this case on the brain. And moreover, you can see that we're predicting the magnitude of the electric field. So that's just how much juice is arriving in each pixel or region of interest. But we can also divide that electric field into a radial and tangential component. Radial means that it's current that's flowing in and out of the surface. So if you have a sphere, current that's either flowing in or out of the sphere would be radially directed, while tangentially directed current would be current that's sort of wrapping itself, it's traveling along the sphere, right? Tangentially along the surface of the brain. And so you can see, for example, in the case of distant bipolar, whereas magnitude is a sort of one big oval blob, under the electrodes, we have radial directed current flow. And between the electrodes, we have tangentially oriented current flow. And that kind of makes sense if you imagine that current is entering at one electrode, crossing across the brain and exiting at the other. Radial current can also be divided into radial in, flowing into the brain, or radial out, so flowing out of the brain. And so you can see we can go from cortical surface radial to cortical surface radial plus or minus, and now we get a sense of directionality. Under the anode, the positive electrode, current is flowing in. Under the cathode, current is flowing out, and it's rather symmetrical in this case. So as far as this first issue of the right tissues, again, the having spheres removes all the detail that we know to be so important about head anatomy. But because it does that, I think spheres are and continue to be very valuable tools in addressing certain questions. For example, if I wanted to compare the role of distant bipolar with adjacent bipolar, if I did that on a real head, I'd have to worry about things like, well, is one of the electrodes moving closer to the orbit? Or is the skull thickness the same under the distant bipolar or adjacent bipolar? So you could see it starts to be a little bit hard to compare apples and apples if I just want to ask in principle what these montage variations do. Or another example would be Uncle Peter Cheff and colleagues published a paper where they compared, I think, dozens of different TMS coil configurations, and doing that in spheres allowed them to do a simple apples to apples comparison. So I think concentric spheres have always played an important role in computational models to ask certain questions, such as relative focality, but we need to also be very much aware of their limitations. And the other point here, as far as point number 4, I'm representing neuromodulation in terms of electric field in the brain, and this is very commonly done. You'll see electric field, you'll see current density. If the surface has one resistivity, like the surface of the brain, these plots look the same. They just will have different kind of labels. Electric field is considered to be a more direct measurement of the strength of neuronal activation. But there's an assumption here that's being made that regional electric field is correlated with degree of neuromodulation, right? Where you see red, where you see heat, in this case, is we're making an assumption that that means more neuromodulation. And sort of implicitly, we're also making the assumption that where you see yellow, which is half the electric field, maybe that's half the level of neuromodulation. And where you see green, that's certainly less. So there's a lot of assumptions caked in here about, for example, a monotonic relationship between electric field and the degree of neuromodulation, which may not be the case. There's also assumptions that every brain region is equally sensitive to the electric field. And there's many other assumptions caked into this as well. And so it's important to keep in mind that when we are plotting electric field, that is already a very intentional or significant assumption of how we want to represent neuromodulation. Now we can go from spheres to more realistic models. My group and others have worked hard on developing these MRI-derived models. And so we talk about gyri precise in the sense that the images, these high-resolution images, represent the folding of the brain cortex, and we develop a modeling pipeline that preserves that resolution. So our first publication here, this is data from that first publication. You can see here the different masks we have chosen to represent, and you see below a plot of how current flows through the brain. One of the things you will see already by eye is that it's kind of punctate. You have these sort of idiosyncratic peaks, and those are direct consequence of the way the current flows through the brain. The other thing you'll see is that we are predicting that the current is not under each electrode, but rather in an area that spans between both electrodes. And in this case, the peak is predicted to occur between the electrodes, not under them. And so here we're asking questions about right tissues. Considering gyri folding is obviously very important, preserving that. But an equally important notion is this notion of CSF continuity. So if you look at the scans themselves at 1 millimeter, it would often appear that the brain touches skull, right? The scans won't have the resolution. And so if you were to just segment it, I would say blindly, but just, naively, you may create a situation where in certain parts in the model, there is no CSF and the brain directly touches the skull. That completely changes the model predictions. In fact, it can create a situation where it appears that the electric fields really are underneath the electrodes. And so in that case, it wasn't just about preserving the resolution in the original scans, but using a priori information, things we knew to be true from other techniques like histology, in order to add additional detail, in order to add additional processing. The other important thing is here is we are explicitly representing the electrodes. So instead of perhaps what was done previously, where you just apply a current to the skin directly, we are actually modeling the electrode itself and applying the current to the top of the electrode. And that also has important implications for what was done. So the fact that we made all these decisions resulted in this type of computer simulation. Now, since then, we and others have worked to add more and more detail, adding more tissue types, gray matter, white matter, anisotropy, both in the brain and so on. And a lot of this work has been very fruitful, but there's this expression I really like, that a difference to be a difference must make a difference. And what do I mean by that? This is an example from one paper where we considered modeling anisotropy. How you model anisotropy is in itself a question mark. There are different imaging sequences that can give you a sense of tissue anisotropy. They don't directly image anisotropy. And so there's post-processing questions about how you go from these images to actual current flow models. So this gets into some of this right tissue issue. And in this case, we're considering both anisotropy in the white matter, but also in the skull. And there's different variations here of a isotropic model, anisotropy in the skull, anisotropy in the brain, anisotropy in both of them. And I'll point you to the reference to unpack the details. Now, by eye, if you were to look at all these different simulations, you would say, well, they're different, right? One picture does not look like the other. But is this difference going to make a difference, right? Are the differences you're seeing between these images meaningful for the question that wants to be asked with the model? And now it depends. If the question was, where should I put the electrodes in order to stimulate this region of the brain? Each of these models may tell you to put the electrodes in the same position, right? So one thing that models are used to do is to recommend what dose we should apply. But if two models, one simple and one very complex, both make the same recommendation of where we should put the electrodes, then I would argue maybe the difference did not make a difference, right? They're both giving the same prescription, but one is much more complicated. And by the way, complicated means subject to many more assumptions. The other thing models can be used for, other than prospectively recommending a dose, is helping, for example, with retrospective analysis. For example, asking, if I compare two individuals who got the same dose, did one receive more brain electric field than the other? But here again, there could be a situation where a very simple model suggests that patient A got more electric field than patient B, and a very complicated model also says that patient A got more electric field than patient B. So the rank order has not changed, even though the specific electric field does. And so again, in that sense, if the goal of the model was to rank subjects by susceptibility to current, the difference did not make a difference. And again, and so I think there's a value, as this paper title says, but there's also a cost to complexity. Now, I do think that obviously in some situations, more detailed models have value. Going back, for example, to this earlier paper, where these gyri precise models with CSF continuity suggested that large pads will not produce focal current flow. This also led us to suggest that maybe using smaller electrodes and maybe putting them in very particular configurations, for example, with a center electrode and 4 surrounds, can produce focal current flow. And so again, it was only because these models suggested that pad tDCS was not focal that we then used them to predict ways that we might do focal stimulation. And both the diffusivity and the focality has since been validated extensively. So in this case, I would say there is a difference, and there is a reason not to always use spheres. And there is value in gyri precise and anatomically precise models. And so if individual anatomical detail matters, then you would also argue, well, then different people should be different. Someone with a very large head and not a lot of folds in their cortex versus someone with a small head and a large gap between the brain and the CSF. The same dose is what we apply on the outside, could produce different electric fields inside the head. And this idea, which we've been thinking, others have been thinking about for a long time, has now been pretty well hashed out in many publications. And in some cases, it's really obvious. For example, if you have a hole in your brain because you had a stroke, and so that part of the brain has not been cannibalized and replaced by CSF, cerebral spinal fluid, is much more conductive than the surrounding brain. And so some of our earliest models looked at the impact of a lesion, and it turns out that current flow is altered by the presence of the lesion. Sometimes in ways you would a priori guess, in some cases in ways that are predictable. We can also look at skull defects. So in the paper cited here, we actually made synthetic skull defects. So we took an MRI and we synthetically drilled holes in different positions in the skull to predict what matters. And of course, having gross anatomical changes and having very resistive tissue like skull or brain, relatively resistive brain, replaced with very conductive CSF will alter current flow. And so that led to the notion that maybe when you have individuals with very atypical anatomy or injuries, dose should be modified for them. But it's not always so simple, you know what I mean? So anatomy matters unless it doesn't, and I can show you a couple examples of that as I wrap up. We also thought about the role of dosing for subjects who may be obese or super obese. For example, work coming out of Harvard where there was interest in looking at the effects of tDCS on craving, food craving. And there's very compelling ongoing work on this topic. But many of these subjects may be individuals who are obese or morbidly obese, and that is manifest also in the fat content in their head. And you can see here scans of subjects ranging from borderline anorexic all the way to very obese. And you can see by eye the variance in the fat content, which is sitting outside the skull. Skulls are the same in this case. The fat is different. And so we asked, well, fat is a resistor. So we thought, well, fat is very resistive, and we're trying to put electrical current into the head. Shouldn't fat impact the delivery of current into the brain in these super obese subjects compared to, let's say, typical subjects? And a lot of detail here. I can refer you to the paper. The shorter answer is not really. I mean, you can look, for example, at subject S1, very obese with the M1 supraorbital montage. We're seeing a peak in the brain of 0.316. It's not that different, or it's not such an outlier compared to the other models with very different BMI. And we see the same thing. So different montages matter. They do matter. And different heads respond differently. But generally speaking, there was no simple correlation between BMI and brain electric field, which is really surprising to us because fat is such a resistor. It seemed like other factors like overall head shape seemed to matter most. But when we dug into it, we realized that there was an explanation for this, and I'll summarize it in 2 sentences. If fat was an incredible resistor, a perfect resistor, then what would happen is that all the current would shunt through the scalp, right? None of it would reach the brain because we have essentially a perfect insulator. And so when we put 2 electrodes on the head, current would just travel from one electrode to the other along the scalp, and none of it would pass through the fat. So very high resistivity indeed blocks current flow. What if fat was a very, very high conductor? If fat was a superconductor, then current would pass through the scalp, reach the fat, and then travel along the fat, right? It's like having a metal shield around the head. None of the current would actually pass through the fat either, because it's a perfect conductor. So having a shell that is a perfect insulator relative to the other tissues, or having a shell that's a perfect conductor relative to the surrounding tissues, in either case would shield the brain. And so then the question is, okay, too high is not good, too low is not good. And it turns out that fat is somewhere in the middle. It's at this point where its conductivity relative to the other structures actually means that having a bit more fat or a little less fat matters less than other things. And so this is something we unpacked. Again, the models allow you to do things like systematically vary fat conductivity to impact the role. And so it's not to say that fat doesn't matter. It's just to say that it doesn't matter as much as other things like skull shape, which would also impact the raw distance between the electrodes. So again, it matters unless it doesn't matter. Here's just one more example. This was a study we did looking at subjects with primary progressive aphasia. So it's neurodegenerative, and there is a meaningful but incremental loss of brain mass in these subjects. And moreover, the type of loss you see is specific to the disease subvariant. And so that's why you'll see these different disease subtypes. We spend a huge amount of effort developing very high resolution images of these subjects. And I should mention, by the way, that there's very sophisticated automated segmentation available that we rely on as well. But as soon as you're talking about atypical subjects, and you're really talking about very precise tissue segmentation to address that atypical anatomy, we often have to go back to more boutique or customized approaches to generate these models. So we generated these models of these 3 individuals. We wanted to ask the question, how is brain current flow altered in these individuals, and can we relate it specifically to their atrophy? So the prediction is that where you have more brain atrophy, the distance between the brain and the skull will increase. And so in those regions, you will get less electric field to the brain, because that increased sheath of cerebral spinal fluid will shunt current away from the brain. So the prediction was that when the brain is farther away from the skull, the electric field will be lower. So we did a lot of analysis related to this. We looked at the electric field across the surface. We looked at the radial electric field, as we've been doing for a decade at this point. And we looked at other metrics, such as what's shown here, the distance from the bone to the gray matter, and we subdivided that into bone to sulci and bone to gyri as well. And the short answer is we didn't see any evident relationships. If you squint, you can believe that you're seeing certain things. It's certainly the case that gyri will sometimes see peaks, but that's always the case. It's also the case that sometimes sulci will see peaks because the current is drawn in. But we couldn't come up with any specific rules. And moreover, the overall patterns we saw in these subjects, while specific to the subjects, didn't seem that different than what we saw in healthy variants, or in healthy adults that were age matched. And what that means is it doesn't mean that these individual subjects' brains and their anatomy didn't impact current flow. It obviously did. You can kind of see that by eye. It's just saying that other things mattered more. For example, things like overall skull shape, or perhaps skull thickness, and other factors dominated the impact of these other variances. And so ultimately, we had to conclude that the current flow delivery to these subjects didn't look that different than the spectrum of what you'd see in let's say, anatomically normal age-matched adults. Doesn't mean that anatomy doesn't matter. It just means that it's simple to. It was hard for us to come up with simple rules of exactly why it matters. It's sort of like everything matters, and therefore no single thing